Move to the next presentation by Dr. Natalia Soriano-Saravia. She is an assistant professor at the Department of Microbiology, Tropical Medicine and Immunology at George Washington University.
Hello, Marta, thank you. You can see my presentation, right?
Yes.
Okay. So thank you to the DC CFAR for the invitation to present my work today. My lab is focused on gamma delta T cells in HIV infection, and my talk today is going to be about their role as cellular reservoirs of HIV infection. I'm going to present both unpublished and ongoing data that is mostly done by Brendan Mann, who is a PhD student in the lab. And here we go. So gamma delta T cells were identified between 1984 and 1986 as a second different lymphocyte population with non-redundant functions with alpha beta T cells. They share characteristics of NK cells and alpha beta T cells. They seem to be a perfect mixture of both, and as such, they link innate and adaptive immune responses. They express T cell receptor, which is composed of gamma and delta chains instead of the alpha and beta that are normally found within conventional T cells. And this is coupled to the CD3 molecules to form the expressed TCR. They are CD4 negative, although we will come back to this point later. And there are some populations that can express CD8. A very key issue with the gamma delta T cells is that they are species specific. And so the lack of a mouse model that allows us to study their immunobiology, the field has been a little slow in that regard. So gamma delta, as I said, they express receptors from both the adaptive and the innate immunity. So for example, NKG2D or TLRs, and they are potent effectors, cytotoxic effectors, and they can exert their function through direct antiviral mechanisms, for example, producing perforin or granzyme B, or an indirect antiviral mechanisms through ADCC or production of interferon and gamma and TNF alpha. But in addition to this direct cytotoxicity, they also have key adjuvant functions, for example, enhancing or promoting DC maturation, CD4 polarization. They increase or enhance CD8 T cell responses, NK cell responses, and humoral responses against infected and tumor cells. So as you can imagine, all these functions are not performed by a single static cell type, but rather there are different populations depicted here in human gamma delta T cells. There are more than these three, but the most characterized are these three: VD1 T cells, which are mostly located in the mucosa, VD2 T cells, which are mostly in peripheral blood and lymph node, and VD3 cells mostly in the liver. And within VD2 T cells, we can also find several different subpopulations, which adds to their complexity. For today's talk, we're going to be focused on these VD2 T cells, which are the best known. We know their specific antigen and most about their immunobiology. So VD2 T cells recognize their antigens their antigen recognition through the TCR is phosphoantigens, which are intermediates of the isoprenoid biosynthesis pathway, which leads to the production of cholesterol, which is here. You don't need to see any of this. Just an example is the intermediate IPP or isopentenyl pyrophosphate, which is produced in this pathway. And we have learned to manipulate this pathway, and as an example of drugs that may be familiar to all of you, statins and bisphosphonates can modulate this pathway, modulating as well VD2 T cell function. So these phosphoantigens are recognized in an MHC unrestricted manner by butyrophilins that present these phosphoantigens to VD2 T cells. So it's not as simple as single recognition by one of these molecules, BTN3A1, but we are learning that these interactions might be more complex than we thought. So anyway, after gamma delta T cells or VD2 T cells are activated, they are clonally expanded as we expected. And this clonal expansion occurs after viral, bacterial, parasitic, or malignancies. So this is the case for most of these infections from VD2 T cells. However, in HIV infection, these VD2 T cells, rather than expand, they are depleted, and we can see an expansion of the VD1 T cells population. So this leads to an inversion of the VD1 VD2 T cell ratio, which is normally found in healthy individuals. And this strongly correlates with HIV disease progression. So the mechanism of this VD2 depletion was reported to be through an indirect mechanism because they don't express CD4, right? So this indirect mechanism was through direct binding of HIV to the CCR5 alpha-4 beta-7 integrin complex, leading to the cell death and this depletion. However, we could see that in untreated infection, there was a transient upregulation of the CD4 expression upon activation. This is data that we published before, and we could see this upregulation just in the presence of IL-2. So from no CD4 expression in VD2 T cells, we could see an increase after activation with IL-2. And we could also confirm this expression in people living with HIV who were very recently infected, less than 3 weeks. We were very lucky to pick 5 of these individuals. And we could see variable expression of CD4 in these individuals up to 22%. So adding to this indirect mechanism, we then added this direct mechanism that was responsible for the profound depletion of VD2 T cells through a direct infection of the cells. So this is an untreated infection. And then what happens after antiretroviral therapy is introduced, so in chronic infection, we know that antiretroviral therapy does not restore normal gamma delta T cell numbers. And there were combined or controversial results about whether or not ART restore VD2 T cell function. So we published this paper last year, Brendan, in collaboration with Matt from UNC from before I moved here. We looked at responses in ART-suppressed HIV-infected individuals for more than, I think it was 1 year. In the end, it was a little more. So we confirmed the profound depletion in HIV seropositive individuals compared with uninfected. And then using a very complex phenotyping that was previously published by our collaborators in the UK, using CD27, CD28, and CD16, we were able to define 4 different populations, which you can see here: gamma delta 28 plus, minus 16, minus 16 plus. And these phenotypes were correlated to the function of these cells. So these 28 cells have a more proliferative phenotype compared to a more cytotoxic phenotype from these gamma delta 16 positive cells. We could not see, however, a difference of expression in these cells when we compare between uninfected or people living with HIV, despite this profound depletion that was seen in the HIV seropositive group. And we could also see no difference in their cytotoxic capacity against DOTA cells between uninfected or HIV positive people. So coming back to the chronic part of the HIV infection, this is what I just told you that leads to a depletion. So our next hypothesis was if VD2 T cells can be directly infected and then they are depleted, the hypothesis that are these cells latently infected and could be a new reservoir of latent HIV infection? So I'm not going to go through the QVOA, which Rebecca just explained, but just differences. We sort VD2 T cells. These are plated in limiting dilution, activated. We used specific activators of VD2 T cells, in this case HMBPP instead of IPP that I told you before. And then after ELISA P24 is performed, we use an algorithm to calculate infectious units per million VD2 T cells. So using this assay, we were able to recover replication competent HIV from isolated VD2 T cells from both people treated in the acute phase of the infection, AHI, or in the chronic phase of the infection, CHI. I'm going to say this because it's very interesting that in some individuals, we had an unusual recovery of this replication competent. In a limiting dilution, the more cells we plate, we should recover more than in lower dilutions. But in some individuals, was not like that. We could see a pattern that was like this here, lower dilutions, higher recovery. And we could see this in around 26% of the individuals. And I'll come to this later. I'm just highlighting this here. So then we tried to look at the contribution to the total reservoir, and this is something that the reviewers made me put in the paper. I didn't want to do it, but they made me. And so we compare what was the contribution of resting CD4 T cells compared to VD2s. But this is not comparable because we didn't use the same number of cells in each culture, although it's normalized to the number of cells that you add or the number of replicates. But you know, the less the cells, the higher the variability. So I think it's not fair. And then the activation we used was not comparable. We used PHA for resting CD4 T cells. So the questions that we had from this paper is, what is the contribution of VD2 T cells to the total reservoir? What is the stability of this reservoir? Is the other subset, VD1 cells, a reservoir as well? What is the contribution to the tissue reservoir, which is very important? And can we reactivate VD2 T cells in a similar fashion to CD4 T cells? I'm going to try to respond all this in the next 5 minutes. This is a paper that we published last year as well, in order to simplify a little bit how we could measure that contribution. The approach, I thought it was going to work very nicely, was from PBMCs from people living with HIV, isolate resting CD4 T cells in the presence or absence of antibodies to deplete those gamma delta T cells, and then perform the QVOA. So using these parallel assays, we could not detect a difference in IUPM between cultures that had those gamma delta T cells or gamma delta T cells were depleted. However, we could see a trend towards a higher infectious units per million cells in those with gamma delta depletion. So when we looked a little closer, we analyzed nadir CD4, peak of viral load peak, you know, all the variables that were in our hands, and we could only detect the higher expression of CD16 in VD2 T cells was correlated with a lower recovery of HIV in those cultures. So this is part of what we are doing also in the lab, trying to see to exploit that VD2 T cell cytotoxicity. And this correlates again. Give me 1 second, please. Give me 1 second, please. I'm so sorry. It was my kid.
It's okay.
So oops. I don't know why this is not okay. So I'm sorry, I lost track. So we then took a closer look at these individuals to see where these dots were going or coming, And this is what we have here, paired by resting CD4 T cells depleted of gamma delta T cells in purple, or resting CD4 T cells with the gamma delta T cells in black. So as you can see, we detected 4 individuals. One was borderline, so we didn't take him, but it was almost there, whose infectious units per million resting CD4 T cells were higher when the gamma delta T cells were depleted. And very important, we took those isolated those VD2 T cells and performed a viral inhibition assay, and we could see that the inhibition capacity of those VD2 T cells from these 4 donors were higher compared to other 3 that were not in that category. This 4 again is around 24 to 26% that we saw in the previous paper. So it's very interesting. That is a kind of a population that is there. So the next question is, how is the stability of the latent reservoir? We know that in resting CD4 T cells is very stable over time. This was work published by Silicano several years ago, and then confirmed later by Margolis. So the time to eradication was around 70 years. So our question was, is the latent reservoir over time in VD2 T cells as stable as in resting CD4 T cells? So this is ongoing work. We used 5 individuals so far. And as you can see, the reservoir is very stable in resting CD4 T cells. However, in VD2 T cells, it's kind of decaying. Sorry? Was that a question?
No, I think somebody has to close the microphone. Yeah.
Okay. So in VD2 T cells, although we see kind of a decay, we need to confirm this, including more individuals. So the next question is, are VD1 cells reservoirs of HIV? We first looked at DNA in Pol HIV DNA, and we could detect VD1s containing that HIV DNA. The caveat here is that this is not intact, so this contains both intact and defective provirus. But we could recover replication competent HIV from them. The next one is about the what about the GALT, the gastrointestinal associated lymphoid tissue? It's important because only a few, a small fraction of T lymphocytes resides in the blood. However, around 40% of them are in lymph nodes, and more than 60% are in the GALT. We know that there's a propensity of the GI tract toward HIV infection with an activated environment and high expression of CCR5. And gamma delta T cell frequencies in healthy individuals can be up to 60% of IELs in the colon. So it's worth it to take a look at their contribution in GALT. So for this, we are collaborating with Mark Siegel and Linda Witkin at MFA, and we are trying to get paired samples from blood and gut from ART-suppressed people living with HIV. We are performing immunophenotyping QVOA, the intact provirus DNA assay that Brendan has already optimized in the lab. And then we want to sequence the virus recovered from QVOA, similar to what Rebecca was presenting. The goal is to have 21 participants, 12 for blood and 9 paired samples with gut, with 1 year apart. So this is just preliminary data that we have. VD1 T cells are the predominant subset in the gut out of the lymphocytes and CD3 positive T cells. We have around 20, in this individual, we have around 25% VD1 cells compared to almost a very small proportion as we expected, and variable levels of CD4 and CD8. This is the immune phenotyping that Brendan is doing currently. We're trying to see what the best way of putting all this data together. But the something that is important to highlight here, and this is just comparing gut and blood, not the healthy with the people living with HIV. This is expected, a higher proportion of VD1 in the colon, represented in gray, compared to the blood. And the other regarding tissue homing receptors in gamma delta T cells, we see very high expression of CD103 both in VD1 and VD2 T cells in the colon compared to blood. And regarding CCR6, which has been reported to have to be a population of CD4 T cells enriched of latent HIV, we don't really see a difference between, well, maybe this, we'll see, we'll have to see. But in blood, at least, or in VD1s, we don't see a difference between them. So what we are going to do or we are just doing, it's QO of the paired blood samples, the IPDA from blood and GALT in paired samples. We're going to look at the compartmentalization of the reservoir. This is something that we did before, similar to what Rebecca was presenting. And we, so far in these 3 individuals, I think we have some more, we couldn't see really that the gamma delta T cells may have a virus that is different from the one that is recovered from resting CD4 T cells. And then we will need to see the response of VD2 T cells to latency reversing agents. In conclusions, gamma delta T cells are a complex T cell population with several subpopulations that, as I said in the beginning, adds more complexity to this subset. So when we talk about them, we need to think outside the box because, you know, depending on the environment, the immune challenge, we will need to open either the cellular, innate, adaptive, or humoral box. VD2 T cells are reservoirs of persistent HIV infection in peripheral blood with several unknowns, starting from the contribution, both in peripheral blood and the tissue. We don't know the stability, and we don't know the response to LRAs. And finally, we don't know whether we know that the VD1 subset from peripheral blood constitute an additional reservoir, and there's some preliminary data because we don't, from Stephen Dix at UCLA, showing some infection of gamma delta T cells in the gut, but they didn't really separate between subsets. So we may assume that that's the VD1, but we need to confirm that. And finally, my acknowledgements. Thank you to the donor volunteers, of course, my current lab, Brendan, Alicia, and Marta, past members, Matt, Chloe, Catherine Cow, all my UNC collaborators, including Carolina Garrido, Nilu Neteleke, Nancy Archin, David Marcolis, Joe Iron, the GW collaborators, Sanjay, Mark, and Linda, King's College collaborators, Dan Pennington and Paul Ryan, who were very fun to work with them. My funding from the NIH, the DC CFAR, and past CFAR and the care from North Carolina. And that's all. Thank you so much.
Thank you, Natalia. Very interesting, the gamma deltas and their contribution in the reservoir. Anybody has a question for Natalia? Okay, so I'm gonna ask a question. Do you think that the reason that you see this inverted ratio of the gamma deltas is because they are stuck in somewhere in the lymph nodes or in the gut or some mucosal sites?
So this is, we don't know. We don't know whether if they are coming from, you know, in the acute infection, there's all the disruption of the gut tissue, and we don't know if they are coming from there, if they are coming out from there, or if they are expanding in the periphery. But I'm very much interested in that because it's like, and I always say this whenever I can, because it looks like there's a specific homeostasis for the gamma delta T cells that they have to maintain a certain ratio, whether it's VD1, VD2, VD2, VD1, but there's always that ratio that needs to be maintained. But that's a very good question. It's very interesting, that part.
In fact. So anybody else has a question for Natalia? Okay, so we can move on. Thank you very much for your participation. It was very interesting presentations. And Sergey, the stage is all yours.
Great. Let me share the screen quickly. Share my screen. This one. Okay, you can see, right? All right, so I'll be very, very quick. I would like to thank everyone for staying that long, and I also would like to thank Marta for taking the burden and helping us to carry this symposium. I also would like to thank Kara and then I think overall administrative core for making this event success. So basically what I'd like to do is at the end, I would like just to remind you that we DC CFAR core, basic science core, which function to provide services and some funding for investigators. So I just wanted to just mention very quickly, and this information also can be found on the website of the DC CFAR. So we have a number of core services, and I think maybe those slides can be also presented there. So services which are marked in bold, these are services that is currently supported by the DC CFAR in part. But basically what we do, we support services which are in demand. So we have labs which can help you with the virology services. We have next-gen sequencing lab at GW. There is a research pathology lab which can be used at GW. There are a number of flow cytometry instruments which I'm going to show you. There is service to characterize reservoirs at GW. There is fantastic microscopy and imaging core at GW. There is multiple parameter flow at GW and also now at Georgetown University. There's proteomics core at Howard. There's also some AEN drug discovery unit which can help you with the drug design. There is a fantastic biocore service at Georgetown University. There's now imaging mass cytometry service, which I think we're going to present in September. And there is also Center for Functional and Molecular Imaging at Georgetown University. So all those services basically to disposition as the member of CFAR. And we also have a number of instrumentations. I think those which are in black were recently acquired with the direct or substantial help from DC CFAR. So we have a number of fax machines, including Fortessa at GW and similar instrument also Georgetown. So we have next-gen sequencing services with single cell 10x genomic service available. We have digital ELISA instrument SPX, which allows you to do this service. So there are a number of microscopes, including transmission electron microscope at GW. So there are some real-time assays that available which can help you to measure viral load at VA. So we just got new Orbitrap mass spectrometer at Howard, which also can be used for number of things, including antibody characterization, for example. So there is also multi-dimensional flow at Georgetown now, and Biocore I mentioned is part of the Biocore services at Georgetown University, and MRI scanners, EEG lab, and near-infrared spectroscopy also available at Georgetown University. So this work requires money, so you can apply for micro grants, so you can have smaller micro grant for a regular service, and also you can have a larger micro grant up to $5,000 for the next-gen sequencing. It requires basically just one-page application, and it's not frequently used, so it's available basically for CFAR users. And also we have pilot programs. So we have pilot for the new investigators, which I think we have now one opening up in September. and it provides $50,000. And then we have bigger funds for the established investigators who are moving from non-HIV field to HIV field, which provides up to 100,000 support. So I think with this, I'm going to again thank everyone and then close the session. And we hope that you will use CFAR services, you'll participate in HIV research, and then we'll see you next year. I mean, in a year, basically, in the same, in the same during the same event. Thanks so much.